Good afternoon. In the first two lectures, I focused on cellular processes, on this dynamic and spatial organization of cells and the physics of cellular dynamics. And with the third lecture, I want to move to larger scales and discuss the collective dynamics of cells, of many cells, in a developing epithelium. That's a two-dimensional tissue. So the subject is material properties and dynamics of tissues. I would like to start with acknowledgements. So this has been work over many years in collaboration with Suzanne Eaton. And I'd like to highlight particular early work by Reza, and then Marko Popovic and Matthias Merkel, Joris Peymans, and the experimental side, Raphael Etonet, Andreas Sagner, Nathalie Dye. So the theme of this talk will be the dynamics, remodeling, physics of epithelia, which are sheets of cells, come in various forms as schematically shown here, but they're essentially two-dimensional manifolds that they define in space, and in most of what I will be saying today, there will be flat, and I will not discuss the three-dimensional nature of their morphogenesis, but focus on two-dimensional projections. And the model system that motivates the research that I'm presenting today is the fly development. And that's sort of the system that was the focus in Suzanne's lab. And in particular, the development of the fly wing, which is a beautiful model system in the sense that is morphologically still quite simple. It's close to two-dimensional, at least in the adult stage. Doesn't have too many complex structures, and it's a beautiful and important model system for morphogenesis and genetics. Now, fly development starts with an embryo from which a larvae hatches that goes through several stages. Instars, where it eats a lot and grows, and then after the third instar stage, it pupariates, and inside the pupa, all the tissues that have been laid aside in the larvae, that have been patterned and that have been growing in the larva, are assembled to produce together to an adult fly, which then emerges from this pupa. So here is a schematical view of the tissues that are laid aside inside, and you've heard this already in previous talk today. These imaginal discs that are precursors of the adult tissues, and the wing precursor is this wing imaginal disc one can find in the larvae. Here you see a wing imaginal disc taken out of a larvae, which, particularly this pouch region, will become the blade region of this fly wing. Here you see also an eye imaginal disc in the process of developing, which later gives rise to the fly eye. In this talk, we'll see later the importance of so-called compartment boundaries, which I want to briefly mention. The tissue can be divided up in different tissue compartments, particularly the anterior-posterior compartment boundary is an important organizer, and cells don't cross compartment boundary lines. There's also the dorsoventral boundary, which forms during wing disc morphogenesis. Here you see the fact that these compartment boundaries persist even to the adult tissue. There is a GFP label of a gene that is expressed in the posterior compartment, and you can see this sharp compartment boundary even at the adult stage, early after hatching in this case. The wing disc grows, starting out from only a few cells, about 50 cells that are laid aside initially, and it grows in about 10 rounds of cell division during this larval stage. 10 rounds of cell division is 2 to the power 10, a factor of 1,000, and you end up with about 50,000 cells after 5 days. And at the end, we have a beautiful fly wing, and characteristic shape is shown here, and below I show you a wing that is misformed because of a mutation in the single gene called DAMPI. Now, one can, since a few years, take out the wing disc from the larvae, culture it in a suitable medium, and then observe the living tissue for several hours. So this is a wing-imaginal disc, followed for about 13 hours in time lapse. This is a TURING GFP, so you see the outlines of cells. You can see cells divide, how cells rearrange. And this essentially is the pouch region which later gives rise to the wing blade. At later stages, in the pupal stage, it is possible to observe the tissue directly inside the pupa without taking it out, sort of an even closer to in vivo condition, by cutting open the pupal case and then watching the tissue through this hole. The wing is sort of close to the surface and one can directly observe it. And roughly starting from 16 hours after pupa formation, one can do that. At the earlier times, cutting the pupa open damages the animal, but after 16 hours, it continues to develop normally, even with this hole. And this is now a pupal wing observed by cutting open the pupal case. Again, time lapse 17 hours. It starts 16 hours after puparium formation. You see it's stitched together from many images. One can see those sort of lines here which will show that there was a stitching process. So it's many images put together into one big image. And then every 5 minutes, about one frame. And one can now observe the dynamics of remodeling of this tissue inside the pupa over about 17 hours in this case. So I want to first discuss some basic concepts we've been using to address the dynamics of these tissues from a point of view of physics. And in this movie that you've just seen, yeah, that's a question. The anterior-posterior compartment is there from the beginning. It is maintained all the time. The dorsal-ventral compartment appears somewhere during the process. Boundary. So the anterior and posterior compartments are there already before when the wing, the imaginal discs are sort of laid aside. Now the tissue is being patterned by gene expression patterns, and these gene expression patterns then later give rise to these vein patterns. So there are patterns of gene expression which are important for the morphogenesis. And this is also partly the theme of the slide here. So we have a very dynamic medium tissue which undergoes waves of cell division. Cells rearrange, cells move, cells divide, cells die. There's active cellular phosphorylation. So this is another example of a two-dimensional active material, similar to what I've shown you in the first lecture. The first lecture was a cellular two-dimensional active material. Here I have a tissue system, but it still has a very similar physics in terms of a thin sheet that is active and flowing. And the dynamics that we've seen is somehow organized with the help of chemical patterns and signaling systems. There are morphogen gradings, there are cell-cell signaling, cell polarity patterns. And this is also an example of a mechanochemical system that is generating dynamic patterns and flows and rearrangements. And it's another example of an active material, where we can ask what are the material properties, and we write down equations that describe the mechanics and the dynamics of such a system. And to start addressing this problem, it's a soft material which is active. There is internal force generation, in particular, for example, when cells divide, there are anisotropic stresses. But cells can also generate, with actomyosin systems with the cell cortex, stresses in the material. And the interesting question are the relevant timescales here. And one can think of this, we have cells in the layer, there is this network of so-called apical junctions which have enforced or stronger actomyosin belt-like structures, which are also the structures that are visible here with this BFP label. One can think of this as a mechanical network that is under tension. And if one uses a UV laser, one can ablate. Yeah.
So, that's important here.
First, I have not yet formulated the model, but I will write an effective two-dimensional model, which hopefully captures everything, but we don't know enough about the properties of the extracellular matrix to know exactly whether we have to modify, where we have to modify the model. So here I'm using a laser ablation experiment, or showing the laser ablation experiment that shows how, if you perturb or ablate a system, there is a relaxation happening. And you can think of there is an instantaneous mechanical perturbation. The system relaxes to a new force balance state. And this relaxation happens in the order of a minute. And this implies we have a broad range of relevant timescales in this problem. And also when it comes to extracellular matrix, this will remodel on longer timescales, presumably. So we can look at sort of the spectrum of interesting timescales. Yeah? If you look at relaxation of local mechanical perturbations, this happens within a minute. But then most of the processes that are relevant sort of for morphogenesis, they happen much more slowly. So cell divisions and cell rearrangements happen on scales of 1 to several hours. And then formation of shapes and patterns is then even longer than that. So that's tens of hours to days, as we've seen that the whole growth process takes about 5 days. And therefore, one simplification one can often use is to think of the problem as instantaneously force balanced, and then as the material properties, as the cell behaviors change on slower time scales, as genetic patterns evolve in time, there is always an instantaneous force balance maintained as the properties adjust, and that is what we call a quasi-static approximation to the full dynamic problem. And I will often use this quasi-static approximation in this talk today, just because it makes the treatment much simpler than trying to write directly dynamic equations which already take into account the short time scales. So one of our favorite approaches is called vertex model, where we are essentially writing an effective two-dimensional model for the three-dimensional sheet, focusing on the observables geometry, which is the meshwork of adherence junctions. But the physics of this meshwork is the physics of the whole three-dimensional layer. So hopefully also including the extracellular matrix. But in the model, it's not visible which elements now contribute to the mechanical properties of the meshwork that we are writing down. And now one of our favorite ways to formulate this problem, and that's a projected two-dimensional version. I think Guillaume Salbreux will also talk about generalizations to three dimensions, is to define what we call a work function. There's an effective potential that describes the work needed to deform this meshwork, this polygonal network. Each cell is a polygon. Alpha is an index of cells. I and J are indices of vertices. And to capture the mechanics of this meshwork, we take into account the fact that there is a preferred area of this two dimensions. And there is an elastic modulus describing area changes. And this comes, of course, from the three-dimensional nature of the problem. For example, in the simple case, one would think of the cell having a constant volume. And by changing the height of the cell, one can change the area of the cell. But in order to do that, the cell has to undergo a shear deformation. And therefore, the K is related to the bulk shear modulus of a cell. That then gives rise to an effective area modulus in the plane. So A alpha is the area of a cell. So this is the area elasticity K. A0 is the preferred area of the polygon. And then we also take into account stresses on cell bonds. So this is like an isotropic stress that is generated here. There's like a pressure in two dimensions that corresponds to this energy term. And the other two terms that describe anisotropic stresses associated with boundaries. And so Lij is the length of the boundary between vertex I and vertex J. This is the sum of all bonds in the network. And lambda would be a tension of this bond. And now in general, one can think of this as an expansion in higher orders of powers of the bond lengths, cut off at a second order. So I'm adding a quadratic term to the linear term. And just because it turns out to add this term provides some interesting features of the model which would be absent if we would cut it off after the linear term. And to not have too many terms here, we focus here on the term in length square, where L is now the perimeter of a cell. So we sum all the lengths of the vertices around one cell and add this sort of elastic term for the perimeter as a term that is generally expected by symmetry arguments or by an expansion, but which has also a physical meaning that is the idea that if the cell area or where the perimeter is stretched, the cell might increase the strength of the tension in order to prevent this from going too far. It is also a stabilizing term if it has a positive coefficient. In my talk, I will always give it a positive coefficient here. And if this is a positive coefficient, then the coefficient lambda can change its sign, which has quite interesting consequences in this model. So this is the cell bond tension, lambda. Lij is the cell bond, and this is the cell perimeter elasticity, and L alpha is the cell perimeter. Now, when discussing this model, an equal sign missing, it is useful to look at dimensionless parameters which characterize the morphology when we look at force balances. And we have essentially four parameters. Yeah? We have preferred area, the area elastic modulus K, we have this tension, and we have the perimeter elasticity. And when we turn this into dimensionless quantities, there are only two parameters which are really important. They can do that by dividing everything by Ka0 squared. The units energy is the same as K times A0 squared. So this is dimensionless. And I can introduce two-dimensionless versions of these two coefficients, which I call gamma bar and lambda bar. And I have to normalize gamma with K times A0 to get a dimensionless number, and lambda with K times A0 to the 3 halves. So these are two dimensionals dimensionless coefficients. And the one thing that one can first look at before investigating the properties of this model is to ask what are the lowest potential states or the ground states of this model. That these are not of direct relevance to the biology, because the system is typically, because of remodeling and cell division and so on, in some configurations which are force balanced, but not ground state, but somehow excited states. But these are reference states, and it helps to understand the structure of the problem. So if we first clarify what are the ground states, then we get a lot of insight in the structure of the problem when we work with this type of model. And here I show you sort of the type of ground state diagram that one obtains if one does that. And in a moment, I will show you how to obtain this ground state diagram. So my two parameters are these two dimensionless parameters. And for each dimensionless parameter, I can sort of characterize what type of network has the lowest value of W for these parameter values. And one finds essentially two regions. One region where hexagonal networks are ground states. And there's a transition line. And then beyond that, networks are typically soft. So here the hexagonal network is like a solid. If you want to deform it as a shear modulus. And down here, there's no shear modulus. So you can very easily deform it. That's why we call it a soft network. And also while here it is regular, here it is irregular in its morphology. And then there is a line where cells collapse, but the whole model breaks down because cells collapse to zero area. That's a forbidden region here for this model. So what I want to do first is to explain to you how one can understand this ground state diagram for this model. As I can do on the blackboard. So I start out from this potential, and I consider the case where all the cells have the same properties. And that's the case for which I want to do the ground state diagram. So a spatially homogeneous system. Then I can write it in the form that I sum over all cells it's the area elasticity, and I can write the tension term using the perimeter elasticity, because I sum over cells, and for each cell, all the bonds together is the perimeter, and I have to divide by 2 because each bond is shared by 2 cells. And then I have also, there's this line tension, that's the perimeter elasticity. And if I now go to the dimensionless form, where I divide by nk A0 squared, then I can write this as 1/2 for alpha. Now I introduce a dimensionless area, A alpha over A0. And then I have simply A alpha minus 1 squared. Then I have my gamma dimensionless P alpha minus P0, where P alpha is now a dimensionless perimeter. Normalized with the square root of the preferred area. And here I've introduced a parameter P0, that's a preferred parameter, which is minus lambda bar over 2 gamma bar. And if you insert this here, you see that it gives you this expression if I also subtract a constant. So this comes from here directly. I've just sort of this quadratic and linear term, I added a constant to be able to write it as a difference squared. Sorry. Is the perimeter of cell alpha. The sum of all the bond lengths that belong to cell alpha. Is it clear? That was Lij, yes. But if you sum over all bonds in the tissue, Lij is the same. So if you sum over all bonds in the tissue over the length Lij, that is the same as the sum over all cells, L alpha over 2. As I've used. Yeah? So, and now I write this as there's a 1 over n missing. 1 over n. I have it. I write this as 1 over n, sum of all cells. Now introduce a dimensionless function E of area and perimeter. And then I have a constant which is unimportant when I minimize. And the function E of A and P is given by 1/2 A minus 1 squared plus gamma bar P minus P0 squared. And what one can do now is first ask, given the parameters lambda bar and gamma bar, what polygon minimizes this function E, and then put these polygons together to a network to have a minimum of the full function W. And it turns out that minima are often highly symmetric, which means they're often periodic tilings of the plane of equal polygons. And therefore, in such a case, it's enough to minimize this for a polygon type and then build a tiling of the plane out of these minimal energy polygons, if this tiling exists. Now, what one can first note is that this function, take here, square. So these are two quadratic terms, A minus 1 square and P minus P0 square. So this sum is either positive or 0. And 0 is the absolute minimum. If this is 0, then we have found a minimum in all definitely. So we can first look for the absolute minimum. A equals 1 and P equals P0. And if we have a given shape of a polygon, then because P comes from a perimeter, behaves like the square of the area. So if you only, and this coefficient gamma is a number which characterizes the shape. And we can first calculate this for regular polygons with n sides. Yeah? And for a regular polygon with n sides, we have P square equals 4n tangent of pi over n times A. That's a geometric property of a general polygon which is equal sides and n of those. And this here would be my number gamma. I call it gamma n. And that's just a geometric exercise to find this value. One can see that n has to be larger than 2 for this to work. N equals 1 doesn't work at all. N equals 2, it still doesn't give you something which has an area. There's a singularity here. And so we have n equals 3 is the first one, which would be an equilateral triangle. And we have n equals 4, which would be a square. n equals 5 would be a perfect pentagon, and n equals 6 would be a perfect hexagon. Of course, we can go to higher, up to n to infinity. In fact, an interesting limit one can take. One takes the limit n to infinity. One can expand a tangent, and one comes to the sphere. Yeah? That has p square equals the tangent. For large n becomes the argument. The n cancels in this limit, and we have 4 pi. See, is this correct? Let's go 4 pi a. Now, we can first ask, where are these perfect polygons solutions in our diagram? And there are solutions if a equals 1 and p equals p0. This means that p0 square equals 4n. Let me just write it with this gamma n. P square, 0 square equals gamma n. And we know that p0 is given here. I think there's something missing. Pardon? What was the question? Gamma is defined here. And for regular polygons is given here. So this means that P0, which is minus lambda over 2 gamma bar, then becomes the square root 2 times the square root of n tangent pi over n. And we can write this as lambda bar equals minus 4 square root n tangent pi over n times gamma bar. And this is a line in the diagram. So if we draw a diagram, lambda bar, gamma bar, which is the diagram which I have also here. Then we have a 0 here. We have a 0 here. That's a line which looks like this. Straight line. Yeah? And now for each value of n, we have such a line. Starting from n equals 3, 4, 5, 6. And then when n goes to infinity, it converges to a limiting line. 3 equals 4, and so on. So now we know along these lines, these polygons are minimal energy shapes. Now we can ask what happens if we go below or above this line. Let's keep n fixed. We can solve this problem for each n fixed and ask what happens in this space. And when we reduce gamma bar, that means that our preferred parameter is increased for this shape. Now the perfect polygons are those shapes with n sides which have the minimal parameter given the area, and therefore the parameter cannot be smaller, but it can be larger, and then we can move away from the regular polygons. By going below one of these lines, we can still have solutions with a equals 1 and p equals p0, but now we have many of them of irregular polygons which have a larger parameter than the regular one. And while along the line there's only one solution, below the line, there are many solutions in a degenerate ground state, but still energy, absolute minima of energy. And that generates sort of non-symmetric shapes that coexist in the ground state. On the other hand, if we move above these lines, then we don't find solutions anymore which are absolute minima. Then we have to find actual minima of this function. And one way to discuss them, one way to discuss them is first assume that we have regular polygons that we are searching for, and then asking what is the size of a regular polygon that minimizes its energy, or we have a fixed shape with a certain value of gamma, and then ask what is the size that minimizes this energy. And this can be done by introducing a scale factor, changing the size of the polygon, and then minimizing a function which I call E prime, which is 1/2. Lambda is my scale factor. It's like a linear scale factor. I have to rescale P linear with gamma, and then I have to scale A with lambda with lambda squared. Now I can calculate dE prime d lambda equals 0 at lambda equals 1. And this gives me a condition for the local minimum condition. And this is a condition which has stable solutions if P larger than P0 and A smaller than 1. And if I take this equation together with this condition, I have two equations for two unknowns, and it gives me A and P. And A smaller than 1 means that there's a tension at the boundary of the polygon, which, like in the surface tension of a droplet, it generates some pressure inside, and therefore the area shrinks a little bit. That's why we need A smaller than 1 and P larger P0 in order to get this tension at the boundary generating a pressure inside. These are stable solutions. While if I change the signs, this would give me unstable solutions, which I'm not interested in. And what this now does is, if I now, let's say, move in this diagram with some color, on a horizontal line, I'm varying lambda bar along this line. I'm starting from the regime where I have an absolute minimum configuration. And then I'm hitting this line where I now have a perfect regular polygon. And beyond this line, at this point, this would be E. So I start out from E equals 0. This here is lambda bar. And there is my point that comes from this straight line, where I have a certain value set by this equation, and then the energy goes up. And I have this for n equals 3, I have it for n equals 4, 5, 6, and so on. And then each time there's a branch going up. So above these lines, I find perfectly regular polygons as solutions which have non-zero energy. There's a limiting point where I reach these solutions at 0 energy, and then I have a branch with 0 energy where I have a degenerate ground state and many irregular polygons of the same neighbor number coexisting as long as they have the correct perimeter. So the next thing I can discuss so these are the lines, n equals 3, 4, 5. I've drawn them here until 6, but you can go to infinity. This is the relationship for a regular n-gon. And now I can show what happens when we reach this line here. So if I walk on this horizontal line, I cross this point that corresponds to one of these points here where I then start to have a regular polygon. And then there is another line here where the solutions collapse. I can also quickly illustrate. And to show why this collapses, I start from my function e of a comma p, and I eliminate the area, assuming I have a fixed shape. And since I'm moving on my branches that go up in energy, I know that I have a regular polygon. So there I can write a equals gamma n, sorry, p squared divided by gamma n, where gamma n is this tangent. And then after eliminating a, I can write a function which only depends on p, which is given by p squared over gamma n minus 1 squared plus gamma bar p minus p0 squared. So here I just replace little a by this expression, and this now is a function which is quartic in p. We have p squared and p squared here, and to illustrate what shape it has, more space. This first term here is a quartic function which is symmetric between positive and negative p. It's just a formal statement. Negative p's are not allowed in my discussion, but just to illustrate it, it would have form of this type. So the plot here is E versus P. And this here is a parabola, which in the case which I'm interested in has a positive line tension lambda, and I want to describe, want to go in this direction here. So this can be either positive or negative. Either I have a parabola which sits this side here, or I have a parabola which sits on this side here, depending on the sign of P0. Now I have to add up these two functions, and what happens, E and P, and now I plot only the positive side. Now if I add up this yellow and this red function, I get something which can look like this. And I have a stable minimum here, but I have potentially a barrier towards this point, which is a collapse cell to zero area or perimeter. And if I now increase this coefficient, I will go via a critical point to a state without stable area. So when I go from here to here, I increase gamma bar for the one particular sign of P0. And the point where I just lose this minimum here, that is the point when I reach this black line. So I have first this point which I explained, and I come to this point, which is exactly the critical point when the finite size polygon loses its stability. And then the model breaks down. So we get this polygonal collapse, which, yeah?
Is there some physical intuition? I don't understand the math.
No, in the tissue, of course, we think of this as corresponding to an extrusion of a cell. Yeah? So we have these collapses happen in the tissue, and you all the time see cells suddenly disappearing from the second dimension, the area goes to zero, and the cell escapes in the third dimension. And that's sort of the remnant of this three-dimensional phenomenon that remains in 2 dimensions.
When that happens, back in
So if this happens to one cell, of course, what I'm talking about now is way beyond what the discussion right now. It's for the whole network when one cell becomes unstable. Here I'm just talking about a single polygon without talking about the surroundings. So what I'm doing here is much simpler than the real problem. But you're asking for a physical interpretation. It's related to cell extrusion.
You varied gamma bar, and then you got this transition. So if you look at the phase diagram, I can independently also vary lambda bar, right? So how do I understand that?
Like here I fix lambda bar.
How did you fix lambda bar?
So in this logic here, I fix lambda bar by P0 and change gamma bar. Lambda bar is hidden in P0.
Lambda bar is hidden in P0. But I can also get a similar transition if I fix gamma bar.
And you can move in different directions.
I can move in different
diagram. I just want to illustrate where this comes from. Because the model is very simple, but at the same time it has some non-trivial properties that one has to extract. So I have now explained all these lines here and this line here, but I've discussed that for a single polygon. And now the question is, can we now use these solutions by just minimizing for a single polygon and now make an argument for a whole network? And for the whole network, of course, in principle, we could put together lots of different shapes, and then we have a lot of possibilities putting them together to tile the plane and then ask what are the minimum energy shapes. So I want to assume that my ground states are regular configurations, except in the region where I have this degenerate ground states. At the moment I move up here, I will compare states of periodic tilings of the plane, and later we can ask, well, do we find other solutions that have even lower energy? And that is not always trivial, and I don't think there's a mathematical proof that they can't exist, and I will show you also examples where they do exist. But the strategy we have pursued is to first obtain all the periodic tilings. And there I want to give you some ideas about how one can identify, in general, the periodic tilings. Of course, you may know them. They're also shown on the next slide. The only periodic tilings that exist are actually those three, and I just want to give you a powerful argument that shows you that this is true. And that is based on the concept of the Euler characteristic that you can define for the polygonal meshwork on a two-dimensional manifold. So we have some arbitrary meshwork, and the Euler characteristic chi is a very simple definition. It's the number of vertices minus the number of edges plus the number of faces. So the faces correspond to the cells. The vertices are these vertices here, and the edges are my cell bonds. And this has the property that it only depends on the topology of the overall manifold on which it lies. This can be actually shown quite easily. Maybe when I add a new edge to an existing network, because I don't change the topology of the manifold. And then what happens is my number of vertices has increased by 2, because I have 2 new vertices. My number of faces has increased by 1, because I have now, instead of 1 face, I have 2 faces. And my number of edges has increased by 3, because I've put in 1 edge here, and 2 old edges are now subdivided, which means there are now these 2 new edges. And then you see, if I have the vertices positive and I subtract the edges, there's no change So by doing any of these type of changes to the meshwork, chi is constant, does not change. And if you remove edges, the same thing happens in reverse. You're just doing this opposite. You don't change chi until you go to the moment where you break the network apart. And that corresponds to changing the topology of the manifold. So the only thing one needs to know by dealing with this is what is the value of chi for different topologies. And if we want to, for example, think of a manifold that is two-dimensional and encloses like a spherical topology, the simplest mesh that has the same topology is a cube. And for a cube, we can now easily count the number of vertices. 4 up top, 4 on the bottom is 8. The number of edges is 4 on top, 4 on the bottom, and 4 in the middle is 12. Number of faces is 6. So this is 2. For everything that's closed like a sphere, we have a value of 2. When we do our calculations with our, that's in respect the correct topology probably for a wing disk, because it's essentially a closed sac that is flattened. But if we do our calculations on a computer, we usually use periodic boundary conditions. And with periodic boundary conditions, you have a box, and the simplest, smallest unit cell would then look like this. So this is one cell, then we have this cell connects your periodic boundary with the one below. So this is number 1, this is number 2, this is number 3, and all the corners are number 4. So there are 4 different faces. So chi is the number of vertices is just 4. Number of edges we have to count is 8, and number of faces is 4. So here chi is 0. And now thinking of such a system which has periodic boundary conditions is most easy because chi is 0. And then we can ask what happens in our polygon network. And I will assume that each cell has a number of neighbors, n alpha, that's the number of neighbors. And I will consider situations for simplicity right now where the vertices have the same number of edges coming in. I call this m, that's the degree of the vertices. In this case, it would be 3, because you have 3 things connected together. And now I can write the Euler characteristic of my polygon meshwork by summing over all cells. Now the number of vertices is n alpha over m. And that is because if for each cell, I sum over all neighbors, then I have a multiple of the number of vertices. And since I know each vertex is neighbored by m cells, I have to divide by m to count correctly the number of vertices. Similarly, if I want to count the number of edges, it's n alpha divided by 2, because each edge is shared by 2 cells. Each vertex is shared by m cells, each edge is shared by 2 cells, and then for each cell I have exactly 1 face. That's the Euler characteristic of the polygonal meshwork, which, if I make periodic boundary condition, is 0. I should erase something. Is that here? And this is only easy to do when m is the same everywhere. That's why I chose this for simplicity. And now we can use the fact that the average of the number of neighbors the cells have in a mesh is 1 over the number of faces times the sum over alpha n alpha. And now, using what is written here, I can write that the average number of neighbors multiplied with 1/2 minus 1 over m must be 1. It's just this equation rewritten. And so we find that the average number of neighbors is 2m over m minus 2. And this defines all the possible cases with constant m. And if I want to have a periodic tiling, which I'm interested in right now, the average of n is the same as n. And then I have n equals 2m over m minus 2. And I have only a few cases possible, because both m and n must be larger than 2 to have some real polygons. So I can look at all possible cases. M can start with 3. With m equals 3, we have n equals 6. These are hexagons, which have threefold vertices. For m equals 4, we have n equals 4. These are squares, which you see here as a second case. If we put in m equals 5, don't find something reasonable, because it gives you not an integer number, so it's not possible. And m equals 6 gives n equals 3. This is the triangular network. And if we would want to go to higher m's, we would have to go to lower n's, which is not allowed. These are all possibilities that exist. And now the question is, now from all the possible polygons that I started from, I have only those 3 remaining. And now the question is, which one has the lowest energy? And that's the one with the best perimeter to area ratio. And the spirit of, if you have a droplet, it becomes spherical because the sphere has the smallest surface area given the volume. Here, of course, the optimal case would be the circle, but circle would have n to infinity, which is not allowed. I cannot make a tiling with circles. So the biggest n that allows me to make a tiling is the best solution. N equals 6 is the hexagonal network. And then I come up with this phase diagram where I have a hexagonal network beyond the n equals 6 line as the ground state. This hexagonal network is non-degenerate. There's only one solution. It has balance between tension and pressure, so E is not 0. And as one approaches this line, the pressure sort of inside goes to 0. The preferred parameter is the actual parameter. And if I cross this line, I have degenerate hexagons. I can deform hexagons slightly as long as the parameter, which is now bigger than the one of a regular hexagon, is satisfied. I still have an absolute minimum, and I have a region of degenerate ground states. And these are then what we call soft lattices. And it turns out that these soft lattices don't have a shear modulus. One can deform them without work, while here one has to perform work to deform them, and therefore there is a shear modulus. This has a shear and a compression modulus. This has a compression modulus, but no shear modulus. And now something interesting happens if we keep in mind those lines. Because as long as I'm between the n equals 6 and n equals 5 line, just a little bit over the n equals 6 line, I have a soft lattice, can deform the hexagons, but all shapes that are allowed have always parameters that cannot be realized by pentagons. This means that I cannot, from the hexagons, create pentagons via a so-called T1 transition, where I have a bond that goes to 0, and I change the topology to go to neighborship relations, where I can go from hexagon to pentagons. This is not possible as long as I'm between this narrow line. But if I cross the n equals 5 line, I can, without performing work, go to pentagons and therefore go through T1 transitions. And that implies that in this narrow region, so on the right side, I have a shear modulus, which implies the energy, if I deform the system, has a minimum and I have to perform work for shear deformation. In this narrow stripe, I have a flat minimum, but then before I can induce topological changes, T1 transitions in a network, I already get a mechanical response. So it's no shear modulus, but always a hexagonal lattice. And if I cross the n equals 5 line, I get a soft lattice where I can do macroscopic deformations, go through T1 transitions without doing any work. Then the energy landscape is really flat. So this is a real liquid, this is a real solid, and in between is something strange, a soft hexagonal network. And so this implies, there's something which was emphasized by Lisa Manning, that the sort of real transition between solid and fluid is not at a hexagonal dimensionless parameter, but by the pentagon dimensionless parameter. So the hexagon, one can calculate it from this tangent pi over 6 in this form, and the pentagon has the tangent of pi over 5, and it's at 3.81. Now, the final thing I want to discuss about this phase diagram is we can now ask, have we maybe missed states that have even lower energy than the ones I've described, where I have a more complex arrangement of unequal polygons? Yes? Yes? So the soft side is completely degenerate. So, so they're different.
A line onto a line.
Yes.
And imagine coming back.
Yes.
The right in.
So because of the degeneracy, and there's no noise here, the energy landscape doesn't prefer any one of those. There would be something like a spontaneous symmetry break. If you add noise, you would get random choices of paths. And only if you're on the right in the green region have you a unique solution, unique state. Yeah. So former student Dr. Staple has searched for these cases, and he found some of them, but in a very tiny region up here. It's if I make sort of a zoom into this region, there is in fact a very small region where you can build states that have lower energy than the hexagonal lattice if you combine triangles with 12-sided polygons or squares with 8-sided polygons. So the squares with 8-sided polygons are lowest energy in this region number 3, and the triangles with 12-sided neighbors are lowest in this region 4. But these are tiny regions. That's essentially negligible diagram. And we don't have a mathematical proof that no other complex solutions exist. Just numerically, we never saw any. Okay, so now I've essentially I don't know. It's an interesting question, yeah. But of course, in a tissue, you probably find it for more complicated reasons than in this, because it's very hard to find this very narrow point in the phase diagram. Because here I talk about cases where everything's homogeneous, where all the bonds are the same. And in a real tissue, it usually is organized that there are patterns. So this was just to explain the properties of the ground state as a reference, that you understand some issues. And now we use that to build a model for a growing tissue.
Frank, do you ever see quasicrystal states, quasicrystalline?
We haven't calculated the energy of quasicrystal. But it must be higher than the hexagonal lattice, because it's n equals, so it exists probably, but with higher energy than it's not a ground state. So the quasicrystal would have 5-fold symmetry, but would not be regular, not be periodic. So one way now to turn this into a model for a growing tissue is to introduce cell division, and I do that for simplicity in a quasi-static manner. So there's always a relaxation of this work function. Fast modes are relaxed, and now a cell can slowly increase its preferred area. This corresponds to volume growth of a cell in the cell cycle between divisions. And then, let's say when the preferred area has been doubled, I introduce a new bond for the cell that now makes one cell turns into two cells. And then I relax this work function again a fast process that I don't describe in detail. I just go to the next minimum. And then I can do that from division to division. And I generate, if these divisions are, let's say, stochastic or randomly chosen, I create a stochastic pattern. This would be, here I start from a hexagonal lattice. Here was already one division happening. This is the number of neighbors with a color code and periodic boundary conditions. And then this is what subsequent divisions that are isotropic in their axis distribution and random from cell to cell generate. So they generate now a stochastic pattern of shapes in the cell packing. If you let it run this for a long time, you get sort of a stationary statistics that is well defined. Now, the energy of this state is higher than the ground state because we perform work when growing cells and when dividing cells. And the system cannot relax because there's no noise here that would allow it to sort of anneal to some ground state. It's a frozen disorder from the divisions. And you see here this energy, the dimensionless one, is a function of the number of cells in such a simulation compared to the value for a purely hexagonal network. Already the first division brings it, or the first few divisions brings it up to a certain value, and then it becomes stationary. You know, the energy per cell doesn't change anymore. This would be the number of neighbors as a statistical distribution, probability or the fraction of n-sided cells from 3 to 8. And one can do that for different parameter values in this diagram, and one gets different statistics, different packings for different parameters. What I just showed you was, I think, this region 1. This happens when we go to region 2. And region 3, this is in fact is in this soft network regime, while the other two are in the hexagonal network regime. And you can see a difference from what the cells look like, and this has to do with this difference, as I'll show you. But you also see different area distributions, different neighbor number distributions, which can be compared to a quantification of real tissue. You see this comparison simulation and measured in the wing disk for case 1, case 2. Now this was chosen already to agree well in its morphology, both in area statistics as well as neighbor number distribution. Case 2 is very different in both ways. Note that from my argument here, the average of n should be roughly 6. And here's the average n measured. The fact that it's not exactly 6 has to do with the subtlety when one does such measurements. And that is, in an experiment, one has a difficulty to counting very short cell bonds because they cannot be resolved in the microscope. So one needs a cutoff length below which one counts a short bond as a vertex. And therefore, we have some vertices which are not threefold but fourfold because there's a tiny bond hidden. And this fact gives rise to this difference to 6. And of course, in the simulation, we have to use the same cutoff as in the experiment to do a comparison. Otherwise, they would not be comparable, these numbers. Pardon? So we go to steady state, and then it's an average in the steady state. So you don't have to call it the time average, it's a steady state average. Now, case 3 is also very different. In particular, you see the area distribution is completely flat in case 3. And this is a direct consequence of the soft lattice. So even though we are having here a situation which is away from the ground state argument, we are essentially creating ground states because in this case, because it's a perfect liquid, it can always relax stress away and can always stay in the ground state. And it's one of these degenerate ground states which is produced. And in the ground states, the area is fixed. lowest energy has preferred area of the model. It doesn't depend on the size of the cells. In fact, because we're on the fluid side in the simulation, we're getting this flat area distribution. What's the question? In experiment, I think biology never has steady states. In experiments, there are never any steady states here. In biological experiments.
I was asking.
In the experiment, we're looking at wing disks at a certain stage and have many of them at the same stage and average. Yeah, and then if you take this case which corresponds to the wing imaginal disk, we can really reproduce the morphology. In the simulation, there is not only cell divisions, but it also generates extrusions. These are the collapses of cells that we talked about before. And even the rate of these collapses matches the ones seen experimentally. So we have about 10 extrusions per 100 cell divisions in the experiment, and we have a similar number in the simulation. So this can really recapitulate basic features of the wing disk. We've also used this approach to discuss the morphology of compartment boundaries, which I introduced before, which are very straight. It's really surprising that they have this organization that remains very straight. And if we take such a model to simulate compartments, we can, for example, label cells blue and red to distinguish the different compartment identities. And if we don't do anything special at the compartment boundary and let it grow, then the boundaries become very rough. And also cells the compartments may even somehow start to break up into pieces. Yes, for example, here you see a little red island in the blue region. And there's no nice compartment boundary. Yeah? And the question now is what maintains compartment boundaries straight and stable during tissue growth? And of course, an idea is that something happens at the interface between these different cell compartments. And if one does laser ablation, one can using labels, one can identify exactly the cell bonds that correspond to the compartment boundary. There's a particular cell bonds for the AP, anterior-posterior compartment boundary. I can look at the rows of cells next to each. And if we now do laser ablation and we ablate bonds at the AP boundary, we get a relaxation that is much more pronounced than if we go in one of these two compartments. So AA or PP would be bonds within the compartments. And there's also the A1, A2, that is one cell row next to the compartment boundary. It also falls down here. So there's a particular set of cell bonds that appear to be under higher tension than all the others in the tissue, and that's exactly the compartment boundary. And so we ask whether, by just making such a change, whether we can keep compartment boundary straight, and in fact that works. So here we increase, we have the same model as before, except all the bonds at the interface have just a higher value of lambda of the line tension. And this can generate straight boundaries. And then we can play with this parameter lambda. That's the factor by which we increase the tension at the interface as compared to the two tissues. And lambda equals 1 would correspond to no change, and lambda equals 4, a fourfold increase. That's roughly what we need to account for the roughness measured in the real compartment boundary of the tissue. So we use a measure of roughness that is borrowed from interface physics. We have sort of the variance of the height variance of the interface shape versus distances over which one measures the variance. And here we vary the value of lambda from 1 to 4, and with value of 4 we get the same order of magnitude for the roughness as in different experiments. So that was sort of a main part what I want to explain about the vertex model. In the last part, and probably I will not finish this today and move it over to tomorrow, the remainder, I want to come back to the tissue for which we have a lot of data, discuss a bit of how, starting from the experimental data of this dynamic remodeling of the tissue, we are building models of the material properties of this tissue. And we are going sort of from a segmentation of such a movie where we can determine the polygonal meshwork, and then we're going from the polygonal meshwork, try to understand how cellular processes contribute to the overall flows and deformations in the tissue. And we want to understand the physics of cell rearrangements. When T1 transitions happen, cells change their neighbors. And then at the end, we come up with a continuum model of tissue dynamics that emerges from these observations. And this vertex model is also useful on the way. So here you see the segmentation using image analysis of these movies. Each cell here is actually a polygon, and we can track cells through the whole movie, several tens of thousands of cells. We can also identify cell divisions. We see when cells get extruded and disappear. If I zoom into this data, you can see the segmentation. So these are the polygons. And in the background, you see the fluorescence of the original image. And each cell gets its label. And then we can track from frame to frame the identity of cells with keeping their label. We can see when cells divide. So this is going to divide. You can see it already starts to round up. Then after division, there are now two daughter cells which get their new labels, which can be tracked afterwards. And so we have a polygonal meshwork where we can apply all these concepts that I introduced. Often it is also very convenient to use a conjugate triangular network. And typically, these polygons are such that they have threefold vertices. And then the conjugate network to get is triangular. So for each vertex, we now have a triangle where the corners of the triangles are the geometric centers of the cell. Each cell bond becomes a triangle edge. Each vertex of the triangle is a cell, and each vertex in the polygon network is a triangle. This would be the triangular mesh. And we can now measure various things. And I would first explain how to measure, how to define a deformation tensor. That when we go from frame to frame, we can measure deformations. I should also do that on a blackboard. Let's see, where's my eraser? This one works. So when we go from frame to frame, we can define displacements of the vertices of the polygons or the triangles, which defines the deformations of the polygons or the triangles. And the vertex displacement is a vector u. It's the positional change of the vertex from frame to frame. And the deformation tensor is a gradient of displacement, a displacement gradient. It's a gradient u, and it can be written as a tensor, as a matrix, b alpha u beta, in a continuum limit. And the idea is, in a polygonal mesh or triangular mesh, how can we define properly the continuum concept of the displacement gradient? And this can be done so that it is consistent with the continuum limit, and it's well defined on small scales. And we use the idea that the displacement gradient, if defined over a certain area, either a patch of the tissue, the whole tissue, or maybe just for a triangle or a polygon, should be something like the area average of the continuum version of the displacement gradient. And if I have a triangle that is deformed by a motion of the vertices, I can just by linear interpolation define a continuous displacement field over the whole area. And it defines me also a continuous displacement gradient. Now, since this is a derivative, and I integrate here, I can do sort of a partial integration and write this as a boundary integral over the boundary of my region I'm looking at. I integrate over the boundary line length. N is the normal vector to the boundary, and U is the displacement at the boundary. That's just
Alpha is not spatial index.
No, I'm sorry.
The spatial index.
This is now a spatial index. Yes, thank you. So yes, exactly. So I should maybe use different indices, but these are now tensor indices for two components in the plane. U is the displacement vector. When you go from frame to frame, so you take the position of a vertex, of a polygon, and then in the next frame, it is somewhere else. And that way you deform the triangle or the polygon. And it's enough to know the displacements of the vertices to calculate the deformation tensor using this rule. Because now what I can do is I can integrate essentially around the boundary using the normal vector and using the displacement vector. And for example, for a polygon, I would just have to do linear interpolation of the displacements along the boundary line and then do the integral around the boundary. And this defines me the tensor. Yes. So let's make a concrete example. I take a polygon for which I've measured displacements of the vertices by going between two frames. So I have three displacement vectors associated with the three vertices. And now I can linearly interpolate in the plane to define the displacement vector over the whole triangle. And that corresponds to this quantity, if I take the derivative of that. This equation is mainly to explain why I can define the displacement gradient by integrating around the boundary. But in practice, this corresponds in this case to an interpolation of the field in the plane. And then I can calculate a tensor that is associated to my polygon. No? This I don't know. I don't think so. I really have to do a line integral around the boundary. I think the definition is the one written here. Yes, here I'm using just kind of mathematics. Yeah. That's the normal vector on each side, unit normal vector pointing outward along the boundary. So I'm integrating around the boundary this product. And this can be done essentially analytically for a polygon. I will, in each boundary, interpolate linearly between the extreme values on the edges. But this sounds a bit artificial, yeah? But this just clarifies the question. But the advantage, the point of this is, if I have a larger patch of many polygons or triangles or whatever, and I define U by the same boundary integral, I get the average of the U's the area weighted average of the U's in here, and I get sort of a coarse-grained version of my deformation tensor. So that's a definition that allows me to define coarse-grained deformation tensor of arbitrary patches, and that is consistent with the definition of the deformation tensor in the continuum limit. So if I can also go the other way around to make these things smaller and smaller, and then I generate the correct continuum limit of the deformation tensor. That's the reason why we choose this definition, but it's a precise definition for each polygon. And the next step then is, the time is almost up, so I could also stop here. Next step, which I do tomorrow, will be to take this general matrix. So this defines a 2x2 matrix, which has 4 different entries. This can be uniquely decomposed now in 3 components: a pure anisotropic shear component, a pure area change component, and a pure rotation. That will be the next thing for tomorrow. Questions before we stop?
Yeah, please. Is there a mic there in your room?
When you are interpolating, ultimately the continuum limit, this displacement or that it needs to satisfy frame indifference. How do you account for that?
Not sure if I understood the question, but one thing, of course, is displacements themselves are not measures of deformation because they are non-zero if you have a pure translation or rotation.
Yes.
And that's not a deformation. So by taking the gradient of displacement, you take away the translations, and you only get something if there is a actual deformation. The material changes its shape.
Yeah. What I mean to say is, if I rotate the frame through which I'm measuring the displacements, that should not affect my measurements of overall strains.
If you do what? If you do?
So this displacement gradient matrix, delta
Yes.
D, which you have measured, if I were to multiply it with some rotation matrix, it would not overall change my measurement of strains.
That's right. So that's why the rotational part here is not strictly a deformation. In this represented, the gradient of displacement takes out translations but not yet rotations. The rotations are here, but you can disregard them. If you just don't consider the rotations, and then this is the real deformation.
It is possible to always write them in an additive decomposition? So here you have used an additive decomposition of the...
Yeah, that's what I...
Yeah, the divergence part is like the eigenvalue.
I didn't have the time to show it, yeah.
And so is it in a small displacement limit where you can only write them additive, or in large displacements also?
So strictly speaking of course, this is defined for a finite displacement. The question is, what's the meaning if it's large? You know? It's a definition, but I guess if you have large displacements, it's more complicated to subtract away the rotations. And you have to do more work. And if it's an infinitesimal, it's just additive.
Good.
So in this, you are taking all the boundaries to having the symmetrical, to be very symmetrical. I'm asking about on one place where you are saying that these boundary conditions are not much possible, where you have one cell which becomes very big and the other surrounding cells are very small. Does to get to that state, can a difference on different elasticity or different properties on different boundary can be, is it important to get to that states?
I'm not sure I understand which part of the talk you're referring to.
So you have conditions where you said that there are very narrow possibilities where you can get a packing where a packing outside near the boundaries where.
I think I know what you mean. Find it. But talk about this here.
Yes. So there on the purple colors, to get to that state, fourth state, is it important to have different, the bigger and the smaller cells to have different properties, or you can reach to that?
Have the same properties. So in all of this calculation, I used exactly the same properties for everybody. And you see, because we're getting close to this collapse line, suddenly small cells are favorable, and by having this combination of big and small ones somehow is even smaller than the hexagonal lattice. And it's something which is surprising. It's not easy to guess.
Because from the first talk you gave, this kind of structures are very much possible when you have a stereocilia or the hair cell patterning in organ of Corti or in other systems. But there each boundaries has different properties.
Exactly. I think here you find it in the extremely narrow region when all boundaries have the same properties. I think you can see such morphologies in tissues, but then the reason is slightly different, and probably the region where they exist is enlarged by making cells different. But that's not the calculation I'm doing here. But it's interesting to see under what conditions one can favor such in much broader ranges of parameters, and then you probably need groups of cells with different properties, or cell bonds patterned with different properties.
During morphogenesis or during the simulations, during the growth, why aren't the cells, deforming in the out of plane, direction, or probably buckling, which might occur due to compression of cell edges or cells?
Is this a question about a theory or about an experiment?
Both. Even in the experiment, we don't see really.
In the experiments, they get extruded in a third dimension, some cells.
Okay.
Leave the tissue, and I mentioned that, and in my 2D model is accounted for by cells collapsing.
Okay, so in simulations, the cells collapse, so that is.
They collapse at the same number as they disappear in tissue.
Okay.
And otherwise, the model is just in the plane, so the model doesn't have degrees of freedoms out of the plane, so that's why it's not.
Okay, so the strains outside the plane, they are considered to be zero.
But you can, of course, generalize this to 3 dimensions.
Okay.
But what I'm discussing here is just the planar version.
Okay.
The questions? Krishna?
Okay, so I'm not sure if my question is super relevant to the system, which is, so this typically solid to fluid transitions have, I don't know if there is a transition here, but typically solid to fluid transitions have a latent heat of fusion and all that and that thermodynamics. Is that particularly relevant here, or when we go away from the ground state that we don't really find these boundaries?
I think formally it's there in the sense that the solid state has a single ground state and the fluid state has degenerate ground states. So if you count the number of states, which is something like the entropy, then there's a jump at the transition. And that somehow corresponds to the latent heat, if you want. And Lisa Manning has looked at this transition in particular as a model for the glass transition also. So it's the simplest model you can use to get the jamming type of glass transitions.
Does that come up in the tissue context ever? Like, for example, if...
I think people think of the tissue when you grow it, the solid state here is amorphous. Also the one that I generate by my growth. So this generates a glassy state. A ground state is a crystal, but it's never realized. So the actual tissue is amorphous, and then the transition fluid to solid is not a normal melting transition, but it's actually a glass transition.
Any other questions? Yeah, one at the back.
Sorry, you've said that the nonlinear term in your W is crucial. I was wondering that what would have happened if you ignored that term and just in a linear regime used it.
You know, the term is crucial to get all the beautiful phase transitions that I've showed. So if you're not interested in this phase transition, this type of fluid solid transition, then you can throw this term away. But this fluid solid transition apparently exists. Yeah? And I think there are now a number of papers who claim to have seen it experimentally. It has been a model for the glass transition, so it is very rich and it has a lot of physics in it. And if you throw away this term, then you lose all of this, and the model is much more boring. But for many situations, it may be enough. So what I will discuss tomorrow, I will only use the vertex model without this term, just because I don't want to go into this transition.
Sure. Yeah. I found a very starking similarity of the polygonal solutions that you have, 3 polygonal solutions. It's very similar to question that we have in math of Eulerian planar tiling. That if you have homogeneous polygons, how can you tile it?
Exactly.
So is the formula, because the formalism there we have just a question of the angle theta that you have, that you just need to make it 360 to make the polygon sit on the plane, and that is what I associate is with the minimum energy, or what ground state are you talking about? So is there a parallel sort of?
And I think there must be a close analogy. And of course, I'm also just looking for possible tilings of the plane. And in principle, I have to look at all possible tilings and ask which one has the lowest energy. And it turns out that periodic or regular tilings are the ideal candidates, but we don't have a proof that these are actually all of them. And it would be interesting to discuss if one can learn something from your approaches.
So in your graph, in line tension versus parameter, there is a small region where both values are positive, right?
Small region?
Where line tension and parameter tension both are positive. Can you?
Which diagram are you referring to?
This graph. Yeah. So after 0, line tension and parameter elasticity both are positive for some small region where the. Yes. So E0 has a negative value.
Both are positive.
So in the small region below there, P0 has a negative value, right? Because P0 is minus lambda bar by gamma bar.
I'm not exactly sure what the question is.
So P0, which you have written on the blackboard, P0, lambda bar by gamma bar, it's minus lambda bar by gamma bar, right?
P0 is positive if lambda bar is negative, yeah.
So there is a reason where both are positive. So P0 would become negative there.
Yes.
What does it physically? I'm not able to understand what.
If P0 is positive, it means that you can have an absolute minimum by having that you have a preferred parameter. If P0 is negative, you can never have the parameter can never be optimal. The system is always under tension. For an optimal preferred, if the parameter is the preferred parameter, there's no tension anymore. And if the P0 is P0 is negative, you're always under tension.
Other questions? Okay, so if not, let's thank Frank for a very nice setup.